All right, cuties, we're going to go ahead and start where we left off this afternoon. So this afternoon, I had a chance to talk to both classes about Tay-Sachs disease and how it's a lysosomal disorder. And so I'm going to move on from there, just still doing the same set of notes. So what they want you to know here is that there are other types of somes, like lysosomes, but these ones are called peroxisomes. And you pretty much don't need to know them, but it helps to have seen the word peroxisome and to be aware that they exist and that there are other somes that exist. So that when the name comes up in a biology exam, which it absolutely could, that you're not like, what is that? I've never heard of that. So the peroxisomes, am I going to test you on this specifically? No, but they break down fatty acids to sugar, they detoxify the cell from alcohol and other poisons, and they produce hydrogen peroxide, which is a very dangerous molecule that has to be broken up. And that's why you have an enzyme that's called catalase, which we will learn about a little bit later. So again, just introducing you to the idea of the fact that there are these other storage molecules that are called that have the ending of the zomes in them. Okay, next are the vacuoles. They are also storage molecule or storage containers. These often contain water, but in animal cells, you have food vacuoles. So food vacuoles contain undigested food, and they digest with they intersect with the lysosomes, and then the lysosomes release their digestive enzymes. So that would be an example of a food vacuole. Contractile vacuoles are really cool. They actually contract, and I love the way they look. So this pretty little critter, it's a protist. So it's like a pond water organism. It's not an animal, but it has this really neat flower-like, I think it looks like a little flower, organelle that's called a contractile vacuole. And the contractile vacuole is actually a pump. And so if this paramecium, it's called, if this paramecium ends up in a water solution where water is entering its body faster than water is leaving, and its body would start to swell, that could kill it. So it has this built-in, super cool, we don't have this, little organelle called a contractile vacuole that pushes water out. Takes energy to do it, so it has to use energy to pump, to make that pump and push water out. And if it's in the other circumstance where water is leaving it, there's nothing that this pump can do. It basically only pumps water out of the cell. So if the cell needed more water, there's nothing that the contractile vacuole can do. And then the other kind of vacuole that is the most important vacuole, arguably, that everybody should know is the central vacuole. And the central vacuole is the vacuole that stores really water primarily in plant cells. Its pressure, it fills, and its pressure pushes against the cell wall of a plant, and it's what helps a plant to stand upright. It's water pressure. That plants don't have a skeleton, so they use their cell walls and this high water pressure to allow them to stand upright. And if you have a plant that hasn't been watered in a while, the central vacuole shrinks, that releases the pressure from the cell walls, and that causes the plant to wilt. As soon as you water the plant, if you catch it in time, if you water the plant, the vacuoles refill, the pressure builds on the cell walls, and the plant is able to stand upright again. The central vacuole also does things like it stockpiles or stores proteins, inorganic ions, et cetera, et cetera. But primarily, I think of it as a big swimming pool in the plant cell, and you've got that. You should always be able to look right at any cell and be able to tell the difference between a plant-like cell, so a cell that has a cell wall, a cell that has chloroplasts, and an animal-like cell. All right, and now we're going to talk about the two arguably most important organelles in terms of energy, I guess, the mitochondria and the chloroplast. So first of all, you definitely need to know the structure of a mitochondria. When I draw a mitochondria, I often draw it as an oval. So if you're trying to draw it in your notes, I draw it as an oval, and then I draw lines in it. So it looks a little bit like a loaf of French bread where the baker has cut slits into the bread. So that's just a way to help you kind of remember or know how to draw it. So there's always the cartoon version, which is on the left here, and then the actual electron microscope version of a mitochondria there. So it has an outer and an inner membrane. The inner membrane is very folded, and that increases surface area, which is actually a super important concept. So although it's small on the outside, on the inside, it's got so many surfaces, and that's where these reactions are going to be taking place, which we'll learn about in another unit. That increases the amount of places where the reactions can occur and increases the efficiency of the mitochondria. So the cristae are the folds on the inside. The matrix is the liquid that's on the inside of the folds, and it contains DNA, which is crazy. Did you hear that? It contains DNA, and it has free ribosomes and enzymes in it. The DNA is a hint at why we think that mitochondria are actually endosymbionts, and I'll get to that in just a minute, so that they came from somewhere other than our own cells. All right, moving on. Their function, obviously, is cell respiration. We call it aerobic cell respiration, which means that oxygen is necessary. There is such a thing as anaerobic. So we're talking about the need for oxygen in order to produce the ATP. So we basically digest. It's called catabolism. We digest our macromolecules that we have eaten, and we break them apart, and then we give a lot of them to the mitochondria, who is then able to build ATP. That's the main energy molecule. And then as a waste product, unfortunately, it makes carbon dioxide and water. Hey, pull this together from your biochemistry unit. You make carbon dioxide. Why is it so important that you exhale your carbon dioxide? Carbon dioxide is very dangerous. Why do you have to exhale it? Give it a moment. What's your blood mostly made of? Water. What happens when you mix carbon dioxide and water? You get an acid, you get carbonic acid. So when the when the mitochondria is making carbon dioxide, it's super important that you exhale the carbon dioxide. It's possible that my dog's going to start barking. People just came in my house. All right, you can kind of hear their voices. Almost all eukaryotic cells have mitochondria, and the more that a cell needs energy, the more mitochondria it has. So if you take a look at this picture, I think it's kind of cool. All these, it says it's stained in red, but in my eyes, these are all orange. But all these little tiny little things, those are all little mitochondria, which I think is super cool. All right, and then another idea that makes us think that that mitochondria are endosymbionts is the fact that they reproduce independently. So the cell might divide, and then at some other point, the mitochondria divide. They're different than our cells. And so, yeah, that's just adding a little evidence to it. So we'll get to that. All right, and then we're going to talk about chloroplasts. Chloroplasts also, like the mitochondria, have an inner and outer membrane. Inside, they have a liquid that's called stroma, and the liquid includes the DNA, which again is weird, right? The mitochondria had DNA in it, and chloroplasts have DNA in them. That's weird because our DNA is stored in our nucleus. Why does the mitochondria have DNA? Why does the chloroplast have DNA? So that's important evidence. It also contains ribosomes and enzymes, and it's where the sugar is made. So remember that during photosynthesis, which happens in chloroplasts, we're making sugar. The thylakoids, if you look at the diagram down below, the thylakoids are the tiny little green discs, and that's where that's where photosynthesis is happening, and it's where ATP is made, and they are stacked into what are called grana. And so you can see a scanning electron microscope picture of the grana, or granum, it's plural, and then each thylakoid is one of those discs by themselves. And what they are doing is called photosynthesis. It is actually building rather than taking apart. So when we're building, it's called anabolism or anabolism. I always have trouble with that word. And it's where we're building sugar out of ATP, carbon dioxide, and light. So the carbon and the oxygen are being recombined into a sugar molecule. Also some hydrogen. Obviously, they get hydrogen from water. There's a source of hydrogen. And it produces oxygen as waste. But then, of course, course, the oxygen gets used by the mitochondria, so nothing really gets wasted. It's just that it gets used somewhere else. And then chloroplasts are what make plants green. I love looking at plant cells under the microscope because most things are not colorful under the microscope. Plant cells are, and they have, the reason that they are is that they have a molecule that's called chlorophyll, which is the primary pigment that captures the sunlight and captures energy from the sun, and it's green. It's reflected back to our eyes as green light. It doesn't absorb green light, so that's why you see green light. That's a weird concept. All right, and then only the plant-like cells have chloroplasts, so no animal cells have chloroplasts. And then finally, energy processing and evolution. Mitochondria and chloroplasts have some crazy similarities. Their jobs are very different, but the way that they have their jobs has some similarities to it. That's a funny way of saying it. I said it awkwardly, but they both are transforming energy. They're both generating ATP. That's interesting. They both have double membranes. Why do they both have double membranes? Both are, in this case, they wrote semi-autonomous organelles. So that means that they are on their own. They have their own DNA. They reproduce on their own. Both of them move, change shape, and divide on their own. They both have small internal ribosomes, circular DNA. You don't know about circular DNA yet. I'll have to mention that. And they make their own protein enzymes. Circular DNA, you guys, is found only in bacteria. What does that mean? What is that telling us? So how can we explain these weird facts and weird similarities? And the idea is the theory of endosymbiosis. So the idea here is that at some point we had very simplistic eukaryotic cells, and we engulfed or absorbed in a bacteria that was, so a prokaryote that was not photosynthetic, and then it just began to live inside of us. We give it food and it gives us energy, and eventually it just became, it always lives in us, it's always there, and it's not, it's never on its own. So the idea is also that chloroplasts were the same thing, that they were single-celled photosynthetic organisms that were absorbed into larger cells and then just became a permanent part of the larger cells. They get protected from the environment, they get fed, and then they provide a service to the cell itself. All right, and then the last thing, I'm not going to say a whole lot about this, but there are other plastids, and just like with the other zomes that we mentioned, these are words that could show up on an AP exam. It's not that you need to know what a proplastid is or an idioplast, but they might show up on the test, and I don't want you to be freaked out about them. They all appear to be products of endosymbiosis, so prokaryotes that were absorbed into a eukaryotic cell, and they have all kinds of different functions. And we have seen actually a similar pattern in algae where certain bacteria, they believe, were absorbed into them, and then that allowed the algae to have certain characteristics that were similar to the prokaryotes. So that would be an example of evolution and how that happened. All right, that is it for the notes, you guys. Let me know if you have any questions. Yeah, that's it. Have a good night.